Welcome to DNA replication. Okay, so you should have watched this video so you understand the structure of DNA. And now we're going to start talking about replication. So to be more specific, we are going through, just a reminder, we're going through the cell cycle. So right now we're going to talk about the S phase. And then later, like next week, we're going to talk about the M phase. So let's do S phase right now. So essentially, when a cell is going to divide, it's going to take its DNA and it's going to make, hopefully, an exact copy of it. So I have this strand right here. If I make a whole new cell, I need to make a whole new strand that has CAT, GTA. If there's a base pair that changes, then I'll have a mutation, and that might lead to something pleasant or not. The evolution will decide. Okay, let's label this diagram S phase. Remember, the S stands for synthesis, which is DNA replication. Cute. So we say DNA replication is semi-conservative. So cute. All right, let's doodle. So if I have a cell, here's my original parent cell. Parent. That original parent cell, if it's one of our somatic body cells, will have 46 chromosomes, okay? So let's just draw its DNA. We're gonna draw it unwound, or no, let's wind it up, right? Here's our cute little DNA strand. When this parent cell divides to give rise to two daughter cells, let's draw them in blue. So these are two different cells. These are my daughter cells. So we just did mitosis. The DNA that was found in the parent cell still exists, so it's conserved. It's just not conserved completely, so it's semi-conserved. The way it would look is this. One daughter cell would get one strand of that DNA. The other would get the other strand of that DNA. So these two strands will split, and then one strand will go into each one, and then it's paired up with new DNA that was made for the daughter cell. So that's how DNA replication works. You take the original strand of DNA, you separate it, and you build in new strands to complement the original strands. So your DNA, like you got one strand of your genome from like that first cell that you were, like that fertilized egg. Some of that DNA from your mother and father still exists in you. It just constantly kind of gets separated and pulled apart. I think that's cool. Okay. So for DNA replication, you mostly have to focus on our enzymes. Okay, if you can remember the enzymes and the order of the enzymes and their functions, then you're going to remember DNA replication. Cool. So let's kind of talk about what we're going to draw here. So we're going to draw a strand of DNA. Cute. When DNA is going to be replicated, you actually will unwind it. So in this two-dimensional, it kind of looks like it's coiled. And so we're not gonna like pull the strands apart. We're just gonna uncoil it and make it open up. So it'll look like this. And we don't do the whole thing at once. You'll get little bubbles. So you might have a bubble open up here. Oh, here comes the cat. Are you here to say hello? Oh, there she is. Cheng. Oh, she's gonna flop. Oh, this is pure entertainment. Are you just gonna be annoying? Okay, you're gonna have to go away. Oh my god, she's so cute. Oh my god. Stop. Okay, I'm going to continue and then I'll kick her off in a second. Oh, the paw. Okay. So it'll open up and then parts of it will still be wound. Oh my God, this is going to become an issue. Okay, you're so cute, but you have to go. Go. Go somewhere. Go somewhere. Go somewhere. Oh, special appearance by Ching. So we still have the same 2 strands. Parts of them are still wound up, but we're ready to replicate this portion. And so we're going to open up the DNA and replicate this portion. So this site is called the origin of replication. Cool. So we're going to originate replication like here at this exact point, right? Like right here on the DNA. And then we're actually going to replicate in both directions. So we're going to have DNA that's replicating that way and DNA that's replicating that way. We'll talk about it. So we have like kind of 2 sites of replication occurring, and they're kind of working opposing directions of our origin, right? So if this is like a super long strand of DNA, to be more efficient, you don't want to replicate from one end to the next. That's going to take forever, right? Your strands of DNA are like meters and meters and meters long. So if we want to replicate a strand of DNA really quickly and efficiently, we're going to open up multiple points of origin. So we're just going to like unwind, 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 and then we're going to replicate in all directions until all these little replication bubbles meet, and you end up with 2 new strands. So right here where the DNA is actively unwinding, so like this little guy right here, this fork in the road, and this fork in the road, those are called replication forks. It is the site of DNA replication. So for every replication bubble, there are 2 replication forks, and they'll be working in opposing directions. Okay, we're going to draw this on the next page, and we're going to draw big. What we're going to draw this 1 replication fork right here. I'll highlight it for you. We are going to draw this. Okay, so we're going to have some DNA that's still wound. We're going to have a fork, and we're going to have a little replication bubble. Here's my zoom in lines. We're zooming in. Zoom. Oh, zoom. That word has such new meaning these days. Okay, I'm going to do it over here because we're going to write stuff here later. So do it over here. Turn your notebook landscape. Let me turn my notebook landscape. And we're going to start with our strand of DNA. I'm going to orient my life right now. Nice. Okay. So we're first going to draw our DNA. Let me draw mine, then you draw yours. So we're going to have most of our DNA will be open, like so. And then we'll kind of go into our fork, and then we'll be coiled up. Oh yeah, that's cute. And then down here, our anti-parallel will be open. We'll go into our fork, and then we're still coiled up. Oh, wow. I did a lot better than I thought. Okay. Let's give some directionality here. So this strand up top, we're going to make this the 3' end. This is muy importante. So this is our 3' end, and you need to follow your strand because the other side will be the 5' end. And then we know our DNA strands are anti-parallel, so this will be the 5' end. Follow your squiggle. This will be the 3' end. Okay, that is important. Now, next, let's draw some nitrogenous bases. So what these lines are representing is they're representing the backbone. So this is the repeating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, over and over and over again. Okay, let's draw some bases in here. So let's do like a few. How far should we space these? Let's just do it. I'm going every 2 lines. Yes. I'm drawing them really short because we're going to be drawing in here, but understand like these would be paired together. We just kind of like unraveled the DNA. Cute. Make sure they match up down here. Once again, keep them short. We're going to be writing inside the DNA. Cute. And then kind of at our fork, they're probably still bonded up, right? So past our fork, they're all still bonded together. So they should be touching. Yes. Cute. Next, let's figure out a DNA sequence. You can literally do any DNA sequence you want. I typically just go random. So let's just go for it. Cute. So once you have your top strand, you need to make sure that your bottom strand is complementary. So if this is a cytosine, this must be a guanine. If this is a thymine, this must be adenine. Guanine, cytosine, guanine, cytosine, adenine, thymine, thymine, adenine, thymine, adenine, blah, 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 blah. So just make sure you follow your base pairing rules. Very nice. Okay. We are going to start with our enzymes. The first enzyme that is muy importante is called DNA helicase. Let's draw. I'm going to draw pretty much all these proteins as blobs, to be quite honest with you. So DNA helicase is found at the replication fork. So this is my fork right here where I've opened up and I've unwound my DNA. The enzyme that does that unwinding is called DNA helicase. So we'll find our helicase here at the fork. Okay. You'll notice our hydrogen bonds were also broken. So here we have hydrogen bonds between our bases and we're wound. Here we're unwound, no bonding. So the reason we have hydrogen bonds between our bases is because they're relatively weak. And so it's easy for this enzyme to come in and break those hydrogen bonds as it unwinds the DNA. If these were covalent bonds, they'd be more difficult to break. It would take more energy to break those bonds. And so we wouldn't be able to replicate DNA as easily. So we have hydrogen bonds there. This enzyme, let's talk about it up here, is called DNA helicase. Cute. What DNA helicase does, and it kind of goes along with this helical structure of DNA, it's a helix, is it unravels the DNA. So it's going to unwind the DNA and it's also going to break the hydrogen bonds between our bases. Break H bonds between bases. And it's a complex of proteins, so it's doing like different enzymes are doing kind of each of those functions. So this helicase is moving in this direction. So it's going to kind of keep moving along, and then as it moves, it'll unwind the DNA and then snip through or break those hydrogen bonds. Very nice. There's another enzyme, and it'll kind of occur on the side after the replication fork. Blob. And it'll be found on the backbone of the DNA. This is called topoisomerase. Ooh, she's a long word. All these things are ending in ace because the enzymes. What topoisomerase does is it's actually going to stabilize the DNA. I have a cute demo. Follow me on this adventure. Where am I going to do my demo? I'm doing it here. So here's my cute demo. These are my 2 strands of DNA. So cute. I'm going to helix. So they're helix. I've been crocheting a lot, that's why I have extra yarn. Cool. So this is like, oh no. It's gonna work. Cute. So here's my DNA strand. My DNA is helical, so it's wound up. So if I have an origin of replication occur right here, and my DNA helicase starts unwinding and breaking those hydrogen bonds, you can kind of see the sides of my DNA that are on the other side of my replication fork get two coiled. So when you open up and when you uncoil one portion of DNA, the other portions of the DNA become two coiled, okay? And when structures become two coiled, they tend to fragment or break because of the tension. So we're gonna have our little helicase here. It's gonna be working in this direction and it's unwinding, but then it's causing this like super coiling action to occur, which we don't want. And so we use topoisomerase downriver of our helicase. And what topoisomerase will do is it's actually going to break the backbone. So it's going to break some covalent bonds in the backbone. It's going to unwind the DNA to release some tension, and then it's going to bond them back together. Okay, so where we had, where's my strings at? Where we like kind of open up and we get this super coiling, topoisomerase would cut one of these strings, unwind it a little, and then bind the string back together just to relieve the tension of this coiling. Oh, such a good demo. Okay, so what topoisomerase does, it's going to break the backbone. Okay, it'll slightly unwind the strand. Not too much, just a little to relieve the tension. And then it'll reattach the backbone. So it's kind of keeping the DNA nice and stable, so it's not fragmenting as we replicate. Cool. So we have kind of helicase and topoisomerase are working kind of in tandem with each other. They're both moving in this direction down DNA. So they're both moving. Everything's moving all the time. So let's draw a little arrow on this buddy. Also moving. Okay, so kind of like these two are occurring together in tandem. Once DNA gets opened up like this, we're going to have our next enzyme come in, which is called RNA primase. Let's draw it like this. So RNA primase will come in, and it's actually going to lay what's called a primer. So I'm going to kind of say, so a lot of replication is hard to draw because everything's moving all the time. So at like time 0, my primase is here. And then like at time like 3 seconds later, my primase is moving. And what the primase does is it builds a primer, and a primer is like a small strand of nucleic acid that kind of begins or starts DNA replication. So I'm going to draw a little bit here. Let's just draw 4. So my primase started here and it started moving in this direction. So it's following helicase. And then let's say at this moment in time, it is now right here. Cute. Let's write about it up here. So RNA primase. So what RNA primase does is it adds a primer, but it adds an RNA primer. We'll talk about that. So it's going to adds an RNA primer. So just think of a primer as like a little starting point, a little piece to add on to. Primer is typically 5 to 10 nucleotides long. So anywhere between there will be a good length primer. Okay. Once we have this primer here, and base pairing rules are going to carry. So if this is a G on my DNA, then this primer will have a cytosine. Thymine will pair with adenine, cytosine with guanine. Here's the special thing about RNA, there is no thymine. So adenine pairs with uracil. So that should be a U. So we have our base pairing rules. And then once we have our primer laid, the primase will exit, and then a new enzyme will come in, and this new enzyme will start building in the DNA to match, right? So we started with this original strand. Now we're trying to kind of separate our two strands and build new strands in between that complement the original, okay? So everything's moving, right? So this primase was moving in this direction until it makes a primer 5 to 10 nucleotides long. And then you'll get your next enzyme come in wherever the primer left off, and it'll start building in its new strand. Okay, so my enzyme would start here at the end of my primer, and it as well is moving. And so let's just draw it down here now. Okay, so this enzyme is called DNA polymerase. I don't have any room to write about this. It's okay, we'll figure it out. So it will follow base pairing rules as well. All right, so we have G, we would have C, A, T, C. So remember it's DNA now, so we're gonna have thymines. I know it's confusing, I know. Okay, so this is DNA polymerase. Okay, let's write about it down here so we don't write too much inside of our little DNA model. So what DNA polymerase will do is it's now going to add DNA to the primer. So we need that primer first. If there's no primer, the DNA polymerase doesn't have anything to bond to, okay? So the reason we start with an RNA primer is because RNA exists as a single-stranded molecule, meaning it can begin a new molecule of RNA from nothing. It doesn't have to attach to something that already exists. DNA is a double-stranded molecule. It can only, and this enzyme can only bond to something that already exists, while this enzyme can start building a strand from scratch. So because of the functionality of these enzymes, we need to start with this little primer, and then polymerase can come in and attach to that work and kind of carry it through. So it's going to add DNA to the existing primer. It's going to be following our helicase here. So as helicase unwinds, our polymerase just follows and then just replicates straight along. It's also going to eventually replace the RNA primer with DNA. Right? So once we've kind of copied this, there'll be pieces of, or different DNA polymerases that'll come, like if this one hits another primer, it'll kind of zip over that primer and it'll replace it with DNA so that we have pure DNA instead of bits of RNA in there. It's also going to be proofreading the work. So we've kind of been talking about these proofreading enzymes when we talked about viruses, RNA viruses, right? Specifically, coronavirus is an RNA virus. And when it does RNA replication in our body cells, we don't have proofreading for RNA replication because we don't do RNA replication. We do have proofreading enzymes, it's these polymerases, for DNA replication. That's why DNA viruses don't mutate as rapidly. Oh, how prevalent. Okay, let's write our last rule down here about DNA polymerase. So polymerase can only add to, only capitals, add to the 3' end of a growing strand. Let's talk about that. Okay, so this enzyme can only add new nucleotides to the 3' end of a new strand. We call it the growing strand. So if we look at our diagram here, I have my 3' end, and then my 5' end would be over here. Because we're anti-parallel, that means my new strand would have opposing directions. So I would have my 5' end here and my 3' end here. So this follows my rule. So my polymerase is going in this direction because it can only add to the 3' end of our growing strand. So because my 3' end is over here and it's in the same direction that my helicase is moving, these two enzymes just follow each other. So as helicase unwinds and unzips, polymerase will just keep replicating and copying. Okay, so we call this strand, this one where they go in the same direction like so, we call this strand the leading strand because it leads the way. Cute. The other strand is more complicado because if you look, my 3' end of my growing strand is going to be over here. And that means it's going to be moving in the opposite direction of my helicase. So how is it going to copy all this new DNA that gets unwound as helicase moves? It kind of jumps. Here we go. We start with RNA primase again. So I'm going to use the same color that I used for my RNA primase here. So my RNA primase is going to add a little primer. Let's just make this a couple. So here's my RNA primer. Here's my primase. All right, same color, color coded. Don't do base pairing rules. It's moving in this direction. Okay, same thing. DNA polymerase is then going to come in and it's going to start adding on to that 3' end. So DNA polymerase will come in like so. We'll just draw the little butt end of that enzyme. And it's going to be adding in DNA bases. Right? So we'll have G, T, C. And it's also moving in this direction. The reason it's going in that direction is because polymerase can only add to the 3' end of a growing strand. That's the bond that it forms. Its active site will bond those substrates. So if this is the 5' end of my original strand, this is the 3' end of my growing strand, and this would be the 5' end of my growing strand. Okay, so now we have an issue, right? So at like this moment in time, when my replication fork was right here, I laid down a primer and I copied my DNA. The issue is my helicase doesn't stop. So that first time when I made my primer, because my fork was right here, I made my primer, I copied my DNA, but right when I copied my DNA, my helicase has already moved. So now my fork is down here, and now I have this chunk of DNA that hasn't been copied. So what happens is another primer will need to get laid. So let's just draw a little 2-piece primer again. So remember it's RNA, so A pairs with U, C pairs with G. We'll have another primase. It'll be moving in this direction. Then you'll have polymerase come in and it will also build in its matching DNA. Okay, so it's also moving in this direction because polymerase can only add to the 3' end of a growing strand. When this polymerase comes to the next primase, it'll just stop replicating. It can replace this with DNA. It'll proofread the work. So now instead of, like up here, we had this nice continuous building of a new strand. That's why we call it the leading strand, because this side gets replicated faster. Down here, though, we're constantly having to replicate a section and then jump. Replicate a section, jump. Replicate a section, jump. Because helicase is moving in the opposite direction as our polymerase. So what we end up getting is we get these little fragments of DNA. And these fragments of DNA, this is my favorite word ever, are called Okazaki fragments. So these little fragments of DNA we get, so I'm gonna do like that. We'll get like this little section of DNA is created, this little section of DNA is created, and they're all kind of not bonded to each other yet. These are called Okazaki fragments. Guess what the guy's name was who discovered this? It was Okazaki. I know, original. So now we have all these little bits of DNA. So we have a special enzyme that will work on this strand only to bond all these bits together. Okay, so polymerase will kind of hit this primer, replace that with DNA, but now we need to bond all of our fragments together. So the last enzyme is the one that bonds our fragments together. So I'm going to draw it as a big old square. Cute. This is called DNA ligase. Where am I going to write this? I'm going to write it right here. DNA ligase. Cute. What ligase does is it binds our Okazaki fragments together. We zoom it in. Binds Okazaki fragments together. So it's the glue. It holds all the fragments together, and then you'll get a comprehensive piece of DNA. So this top strand I have, this replicates DNA super fast because it's going in the same direction. This one replicates DNA much slower because they're working in opposite directions. So if this is our leading strand, then we call this our lagging strand because it lags behind the other. Cute. So that's essentially it. Let's kind of summarize our findings. Go back here. Okay. Last thing I kind of want to talk about. I don't want to confuse you, but back to this guy. This is my origin of replication. So this is the first point helicase bound, and you'll have actually multiple helicases. And so you'll have one helicase moving down here in this direction. You'll have another helicase moving down here in this direction. So we're going to be replicating in both directions. Here's the kicker. So in this diagram we just drew, we said that this top strand was our leading strand because it ran from 3' to 5'. So that new strand was 5' to 3'. We said this was our lagging strand. Cute. So remember, our leading strand is being replicated in this direction. Our lagging strand is being replicated in that direction, but in little fragments, little bits, little pieces. When the lagging strand hits this origin of replication, everything changes, right? Because this helicase is moving to the left in my paper. This polymerase is also moving to the left. Once you hit this origin of replication, this isn't the lagging strand anymore. It becomes the leading strand because now it's going in the same direction as the helicase. So it'll switch. And then this other one would be the lagging because it's moving in the opposite direction as the helicase. Okay? So you can't identify like, oh, this strand is definitely the leading or the lagging strand. This is that. You need to see where your origin of replication is. You need to see the replication fork in which direction helicase is moving. If replication is occurring in the same direction as helicase, that's your leading strand. If it's occurring in opposing directions, that's your lagging strand. But it's going to flip once you hit this origin of replication because your helicase is now moving in the other direction. Okay? Yeah, that wasn't confusing. Okay, so things you should know and be able to do. You should know the enzymes and you should know them in order. So maybe to quiz yourself, I'm going to list the enzymes. You should be able to tell me the function of each of those enzymes. Okay, so the first enzyme, we will kind of in tandem, right? But we used DNA helicase. Okay, remember that one unwinds the DNA and then it will break the hydrogen bonds. I'm going to leave some space because I want you to fill in, quiz yourself, fill in the functions of our enzymes. Kind of in tandem with that, we have topoisomerase. Remember my cute little yarn demo? Yeah, how could you forget? Then we have RNA primers. Remember, it's RNA. We're laying an RNA primer because RNA primers can begin a new strand from scratch, whereas DNA can only add on. So remember our fourth enzyme, DNA polymerase, can only add on to an existing strand of nucleic acid. Okay, and then finally, for just the lagging strand, we'll have DNA ligase. So that'll be that glue that bonds all of our Okazaki fragments together. Cool. All right. Review, go take your quiz. I have a meeting. I think I said this in my first video this week. Friday, so tomorrow, I have a meeting and we're gonna decide like all our grading stuff. So I've been holding off on grading things just because we haven't decided what earns credit or what goes in as like for completeness versus not completeness, stuff like that. So we're still getting all that figured out. So I kind of held off on grading stuff. I will let you know after my meeting on Friday what's going on. In terms of late work from past weeks, or if you're getting behind, you can turn in anything late. I'm not going to dock any points for late work because this is a stressful time and I really just want you guys to be taking care of yourselves. And if that means that you put off a quiz for a couple days, I'm not going to get mad at you. So just get the work done, like stay engaged, and you're going to be fine. I really don't want you to be worried about a grade in this class. I want you to be more focused on learning and staying engaged with the material. So let me know if you need me to unlock a quiz on AP Classroom and I can unlock it for you. I don't think we're going to be able to do retakes, so I'm sorry, but I also think that the grading scale is going to be really, really generous. So I don't think you're going to need retakes. Other than that, thank you for being amazing. Sorry, I've been like so mushy today because I miss you guys so much. Thank you for staying engaged. Please let me know if you need anything. We're going to start doing a lot more FRQs just to get you guys practicing. I want to do timed ones so you guys start writing them in really, really, really quickly and efficiently. And then I'll be giving you really good feedback for those. Okay, I miss you. I'll talk to you later. Bye.